integrating current analyses of the breast cancer microbiome. So this project integrates the 4 available studies related to breast cancer and the mammary microbiome to reassess the original findings in light of the advances in this rapidly progressing field, and to incorporate all the data available as a large meta-analysis to identify general trends and specific differences across the patient cohorts and studies, where the 2 questions our research will address are: 1, how robust are the original findings to the reanalysis with modern tools? And 2, are there new biological insights about the disease that emerge from a combined meta-analysis of all available studies? In this poster, I am presenting my results from the Data 2 analysis of the data from the Urbaniak paper and the Haken paper, where these 2 papers differed in their patient sample size, the bioinformatics tool used, the reads, and the variable region, where our workflow includes pre-processing the reads and then running the data through Data 2 and FileSeq, and then compare the Data 2 findings to the original studies, after which we do the phylogenetic analysis and then move on to the PCOA analysis, and then integrate our findings from each study into a meta-analysis where we will implement multivariate analysis and random forest application. So the results from the Data 2 analysis of the Urbaniak paper and the Haken paper are that in figure A, we show the bacterial composition at the genus level from the Data 2 analysis of the Haken data, which includes Aphasia, Bacillus, Choanozoa, Pectobacterium, Serratia, Streptococcus, and Streptophyta. And at figure B, this is the results showing the bacterial composition at the genus level from the Data 2 analysis of the Urbaniak data, which includes Escherichia, Shigella, Flavobacterium, Lectospiraceae, Mesorhizobium, and Salmonella. And figure C and D show a comparison of the data from the original study compared to the results from the Data 2 analysis of the Haken data. So figure C shows an abundance plot from the original Haken study showing the bacterial composition of the benign and cancer samples at the phylum, family, and genus levels. And figure D shows an abundance plot from the Data 2 analysis of the Haken data showing bacterial abundance of the benign and cancer samples at the phylum, family, and genus levels. The bacterial taxa in both figure C and D don't overlap at any taxonomic level and do not contain any similarities. So these results show that our findings are different from the original studies. And this is exciting to discover that our preliminary findings do not align with the original results, as this means that there are biological factors in the updated datasets, so SILVA, that no one has explored yet. The Haken paper uses the IM Tornado and Illumina-MiSeq approach in their bioinformatic analysis, and my approach is to analyze the Haken data using the Data 2 software and the SILVA reference database, and my approach is a more updated and recent approach, which will result in more accurate results. Thank you.